Good afternoon, everyone. I'm Greg Gilligan. I'm the Vice President of Operations here at RVA 757 Connects. Thank you so much for joining us this afternoon for the latest edition of the RVA 757 Connects Virtual Innovation webinar, Spotlight webinar. This afternoon, we have a very fascinating program about Lyme disease. Sounds interesting. Yes, it is. We will hear from the inventor behind the leading canine Lyme disease vaccine, and he and his team are also building on that success by developing a safe and effective human vaccine. So we'll hear more about that in just a moment. Before we begin, I just wanted a couple housekeeping matters. We'll hear from our sponsor, Horrigan. Then we will give you an update on what's happening with RVA 757 Connects. We'll dive into today's topic about Lyme disease vaccinations. Then we'll leave some time at the very end for some questions and answers from you. As a reminder, we record all of our conversations, our webinars, and this recording will be available this afternoon on our website for you to view and to share with others. If you have any questions for the two speakers, please use the chat function and we will get to them towards the end of the program. RVA 757 Connects is a nonprofit organization, so we rely on donations for our operations. Here is a list of our corporate supporters. We appreciate and recognize them for all their help and support. One of our corporate sponsors is Horrigan. That's the fully integrated construction and development firm based here in Richmond. Here more to tell us about Horrigan is Brian Jones. He's the president of construction division of Horrigan. Brian joined the firm in 2016 and has led strategic planning efforts, oversaw enhancements to the workforce culture. Brian also serves on the board of RVA 757 Connects and also serves as a member of our executive committee. Brian, let me turn it over to you and tell us a little bit more about Horrigan.
Thanks, Greg. Really appreciate it. As Greg said, that Horrigan is a construction management and development firm. We, similar to RVA 757, do work all along the entire I-64 corridor. So we have a lot of wonderful projects going on in the Hampton Roads region, Richmond, Greater Richmond area, and then Charlottesville and heading further west. Variety of project sizes really depends on the client needs and the areas, the markets we serve are mission critical and data centers, healthcare, higher education, advanced manufacturing, office, some commercial and federal. Just span a wide spectrum, some multifamily as well, project types. And I know thankfully, there's several wonderful clients that we serve on this very call and appreciate the opportunity to support them and what they're trying to accomplish. I also appreciate the opportunity to support RVA 757 and the efforts that Greg and John and team are making for the furtherment of our region in a variety of ways that we're all going to reap the benefits of their efforts. So, Greg, thanks again. Really appreciate it. And looking to learn a little more about the breakthroughs with Lyme disease and how we can all be better equipped to handle that. So thanks again, Greg. Appreciate it.
Thank you, Brian, too. And thank you not only for your service on our board and our executive committee, but to Horrigan for its financial support, and also to Mark Horrigan, who served on our board before you did and stuff. And on our executive committee. So we appreciate the support that we get from Horrigan. Let me now talk about a little bit more about what we do here at RVA 757 Connects. Our mission is to improve the economic success and quality of life of everyone in the Richmond region, which we call RVA, and Hampton Roads, which we call 757. We are a coalition of top leaders representing businesses, higher education, and organizations. We are following a national model where nearby localities collaborate on initiatives that advance both regions. We are advancing the mega region from the Metro Richmond area to Hampton Roads as the I-64 Innovation Corridor. As you can see here, we're pursuing several different priorities. We are helping the I-64 Innovation Corridor become a global internet hub. A little bit more on that in just a moment. We're advocating for multi-use trails such as Trail 757 and Hampton Roads, and the Fall Line Trail here in the Richmond region, and the Virginia Capital Trail from downtown Richmond to Jamestown, and now an extension of that trail from Jamestown into Williamsburg. You can read more about our initiatives and our priorities and our other events that we're doing on our website, rva7x7connects.com. One of our priorities is to encourage and showcase innovation. And we're doing that in a couple of ways. One is hosting these innovation spotlight webinars. Mark your calendars for our next webinar on Wednesday, May 6th at noon. Be on the lookout in about a week or so for the topic of next month's webinar. Our top priority is to spearhead efforts to have the I-64 Innovation Quarter designated as a global internet hub. Our plan is a bold and innovative approach to harness a once-in-a-generation opportunity to transform the future of the Richmond region and Hampton Roads. Let me give you a quick update on what's currently unfolding before the Virginia General Assembly and the proactive steps that our organization has and continues to take to ensure policymakers understand what's truly at stake. Digital infrastructure, including data centers of all sizes, is a foundation to our mega region's competitiveness, and this is a pivotal moment. The General Assembly ended its session a couple of weeks ago, but it didn't finalize the state budget. That's because there's a big debate over the exemption on sales and use tax on data center equipment. Virginia has had an exemption for the past 15 years when the General Assembly said, hey, let's have a digital hub here in Virginia. And that really has paid off, especially with Northern Virginia. But now we're starting to see data center expansion throughout Central Virginia, Metro Richmond, and other communities around the Commonwealth. The tax exemption currently expires in 2035, but the Senate wants to end this exemption on sales and use tax on January 1 of 2027, less than 9 months from now. The House of Delegates wants to extend it to 2050 with some environmental provisions. Digital infrastructure runs our economy, and it's what our businesses needs and our universities need to have to be competitive on a global stage. So what we've done is really doubled down on this and said you need to think about data centers as much as a bigger story. It's all about digital infrastructure ecosystem. And this ecosystem, by all accounts, is driving growth and investment growth and really giving Virginia a competitive advantage and our mega region like a competitive advantage. And what we've seen is that mature infrastructure markets or digital infrastructure hubs have better story in productivity per worker, and productivity per worker really drives that local GDP growth. What we're saying is let's not eliminate the sales and use tax exemption on data center equipment. If lawmakers do so, then the next wave of investments is likely to go other places. I want to go a little bit more detail into that. Our business case is for policy change. This was put in place 15 years ago that the exemption would be lifted in 2035. You just can't change a business deal in midstream. It really damages our state's reputation for stability. The other thing is that our economy really depends on this continued digital infrastructure growth. We need to keep it going. The last couple of years, 84% of the total capital investment in Virginia was in data centers. It really has been the juggernaut and something that we just don't want to give up or stop abruptly. The issue comes down to fair taxes and how it relates to a particular industry. This particular tax is on equipment, and it might have been appropriate several years ago when you built a data center and that initial equipment would stay in place for 7 or more years. However, because of AI and just the computing power of the equipment, data center operators now purchase equipment and refresh that equipment and just way beyond what anyone had ever appreciated would be the understanding of what this tax would do. This tax actually is taxing the engine that's driving our economy. Most of the other 35 states that have this exemption, which comes with some type of thresholds that you have to have a certain type of investment or a certain number of employees that are going to happen there in order to get this exemption. As I mentioned before, this business model has shifted the pace of technology and shortened the refresh times, meaning you're taxing equipment that it isn't staying in place for as long of a period as it was just a few years ago. It's just a lot different these days. So what could happen? We think expansion would slow first. You wouldn't have the mass exodus of data centers, but expansion slow, upgrades, and refresh recycles would be next. We become a mature secondary market, and long term, our footprint shrinks. What we built over the last 15 years is in jeopardy, and we believe to protect this investment and really continue to nurture it, we need to do that. So what are we, RVA 757 Connects, doing, and what have we been doing the last couple of months? We launched an online digital infrastructure and data center information resource hub. It's just about all the what and the why and the how of digital infrastructure and data centers to really explain the arguments behind this industry and just how much it impacts businesses. We also created a series of videos with top executives talking about the impact that the data center industry brings to Virginia beyond the data centers. Let me play one of these for you.
Data centers are just one part of Virginia's growing digital infrastructure ecosystem that sparks ripple effects throughout the economy by adding new jobs, fueling innovation, and driving growth across the state. In this series, we're taking a look at the people and businesses that lead them.
So Acoustical Sheet Metal Company manufactures packaging for backup power. And that's making sure that if the lights go out, you have a generator there to power the data center and really get things back up and running in seconds. The best part of this journey has been the growth of our team. Back in February 2019, when I joined the company, we had just 100 employees. Today, we've grown to more than 650, and we're not stopping there. We're excited and proud that Acoustical Sheet Metal is expanding its footprint in Virginia Beach for the third time in 6 years. This time, we're investing $50 million to grow our operations and create 350 new jobs. We believe that this industry is very robust. We play an important part in it, and that's what gets me up each morning, is to continue to reach for the stars.
This supply chain story is going on all across Virginia and certainly across our mega region. So we encourage you to go to our website to see more of the videos that we have there. We also reached out to General Assembly members, and we've shared our new vision map on what we're trying to build as a global internet hub. We crafted op-eds and sponsored content and other outreach media efforts. For instance, we had an op-ed placed in the Richmond Times-Dispatch in early March, both online and in print. We sponsored Virginia Business email blasts. We've sponsored 2 days of Virginia News site to push out our content. We placed sponsored content with Richmond BizSense. We sent out another packet to members of the General Assembly that included a thoughtful white paper trying to make the point that the exemption is slated to end in 2035, and it's codified in law, and let's let it run its course. And let's support the industry that really has put Virginia on the map and really can work to improve our productivity per worker and help us leapfrog back to our leadership in economic growth. You can help by visiting our website and look at these op-eds and white papers and download any of them and send them to your friends, particularly those in the General Assembly. Also, take a look at our digital infrastructure and data center information hub to learn more about this industry and pass along the information to others. So now let's turn to today's topic. We're going to hear from a researcher at Virginia Commonwealth University, who's one of the nation's leading experts in Lyme disease. He's the inventor behind the leading canine Lyme disease vaccine, and he and his team are developing a safe and effective human vaccine, applying decades of research to prevent this tick-borne disease before it starts. Lyme disease is the most common tick-borne illness in the United States, and if left untreated, it can lead to serious long-term health problems, including nervous system issues, heart problems, and joint pain. Lyme disease cases are increasing as ticks expand into new regions. The disease affects public health, healthcare costs, and workforce productivity, making prevention, research, and early detection especially important. So we have two people are leading this effort. First, we will hear from our two speakers today. First will be Evelina Machiva. Hopefully I mispronounced that, which I think I probably did, didn't I, Evelina? She's the VCU's Assistant Vice President for Innovation. She will give us an overview on how research and discoveries made at universities have been licensed to businesses and entrepreneurs who turn them into commercial products, and how universities are functioning as engines of growth. And then we'll hear from Richard Marconi. He's the inventor of the Lyme vaccine for dogs, but his official title is professor in the Department of Microbiology and Immunology at Virginia Commonwealth University. Before I turn the program over to them, just as a reminder, if you have questions, please use the chat function, and I will get to those questions at the very end. So now I'm going to stop sharing the screen and turn it over to Evelina.
Thank you, Greg. Hello, everyone. I would like to tell you quickly about VCU and VCU Tech Transfer and Ventures. VCU and VCU Health are in downtown Richmond. We have 14 colleges and 3 campuses, over 200 programs. VCU Medical Center is the only academic medical center in Central Virginia. You can see the numbers. How they progress. All right. VCU and VCU Medical Center are one of the biggest employer in the Richmond region. As you can see, more than 95,000 jobs, more than 18 billion in economic impact for the Commonwealth of Virginia, and more than 8 billion economic impact to the Richmond region. Our research funding has doubled in the last 7 years. You can see that we're over 580 millions in sponsor funding, and we're ranked number 6 among nationally among public research university by NSF. A little bit about my office. VCU Tech Transfer and Ventures is responsible for protection and commercialization of innovation developed by VCU faculty, staff, and trainees. When VCU members develop new technology, they send an invention disclosure to our office. We file for patent or copyright protections. We validate and mature the technology, and then we market it and license it to either existing company or to new venture. In addition to IP protection and commercialization, we support new ventures, and I will tell you a little about that. We provide new source of research funding for our faculty, and we promote industry engagement. Why we're doing that? What is the value of translational research and technology transfer? We do this for a public benefit, to move the discoveries to the private sectors so they can benefit the public in terms of new drugs, medical devices, or consumer products. They help us recruit and retain talented faculty, postdocs, and graduate students. We bring funding and prestige to the university, licensing revenues, and through establishing new ventures in partnership, we support the regional economic development. These are some numbers of the impact from our office. You can see that close to 100 new startups were spun off the last 10 years, 45 plus million in licensing revenues, and more than 60 new products were introduced to the market. You can see here one of our latest invention in the market. This is a nerve tape for sutureless repair of injured nerves. It was invented by our orthopedic surgeon, Dr. Jonathan Isaacs. It was introduced in the summer of 2024 to marketplace after FDA clearance, but is becoming gaining very rapidly market shares and probably will become one of the biggest VCU money generator. A few other products developed at VCU. You can see medical devices, clothing agents, fashion, consumer products, software. So a variety of different products coming from different parts of the university. How do we support startups? We coach and mentor faculty entrepreneurs. We have an entrepreneur in residence program. These are experienced serial entrepreneurs, industry experts who provide face-to-face advice and coaching to faculty. We support SBR, STTR grants. We help company recruit leadership teams, CEO suite, and create advisory boards. We help with marketing, website creation and communications. We help them reach investors, advisors, and different types of funding. And we do networking events. We connect them with the innovation partners in the region and in the Commonwealth. And here you can see an example of a recent VCU startup, Evolira, which benefited from our entrepreneurship and residence program. This is a company focused on treating of muscular dystrophy. Few more others VCU startups. Interesting fact is most of the VCU startups stay in Virginia. 77% of the new ventures in the last 10 years are Virginia-based, and more than 80% are still in business, which is a pretty good statistic in terms of survival of new startups. We also help promote industry engagement. We target industry that align with VCU core research strengths. We use matching platforms that are AI-based to reach the right companies. We coordinate reverse pitches, innovation stories, visits of companies to town. And here is the team. We have only 3 licensing managers, which makes us one of the smallest offices in among our Virginia peers or national peers. But we're doing quite well. We're number 1 in the Commonwealth in licensing revenues, and we're number 1 in startup creation. And in the end, I would like to introduce today's speaker, Dr. Richard Marconi. He and his team created the Lyme disease vaccine for dogs, which is the biggest money generator for VCU currently. We funded the technology with about $250,000 from our commercialization fund. But in addition, Pfizer Animal Health, now Zoetis, put more than $8 million in sponsor research. This shows the involvement of technology transfer and licensing into the bringing industry sponsor research to universities. Entered the market in 2016 and rapidly gained market share. Right now, it's the largest market share in canine vaccines, and generated more than $18 million in licensing revenues for VCU so far. With it, I will turn it to Dr. Marconi. I'll stop sharing.
Hey, thank you, Evelina. Okay, so I guess as they used to say on Monty Python's Flying Circus, now for something completely different. So what I'm going to do today is give you a little background about Lyme disease. And I think for you to understand the significance of Lyme disease in the Commonwealth of Virginia, it's important to understand a little bit about what causes it and the ticks. So I'll give you a little bit of biology, and then I'll close by talking a little bit, without getting too technical, about the unique aspects of the vaccine that we've developed and how we're advancing this for developing other vaccines. So Lyme disease, as you know, is a very important disease that affects people across the country. And it's caused by a bacteria that has a very unusual name. The name of the bacteria is called Borrelia burgdorferi. And that bacteria is shown in a video taken in my laboratory. The name Borrelia burgdorferi, if you're wondering where that comes from, I had the pleasure of doing my second postdoctoral training stint at Rocky Mountain Laboratories in Hamilton, Montana, which is a National Institutes of Allergies and Infectious Disease NIH lab. And I had the pleasure of working with a gentleman named Willy Burgdorfer. Willy Burgdorfer is credited with the discovery of the bacteria. So the bacteria was named after Willy, and that's where Borrelia burgdorferi comes from. So if you look at this bacteria over here on the right, you'll notice a couple of interesting features about it, which help explain the disease process. First of all, it has this spiral structure, and that's why we call it a spirochete. But that spiral structure is really important in the disease process because when these bacteria transmit from a tick into a mammal, they have to very rapidly drill through tissues in order to cause an infection. So you can think of a spirochete like a corkscrew, and it enters into the tissue and it drills. And in humans, we can often see this manifest in the form of what most of us would refer to as a bullseye rash. Well, the reason that that rash develops is because the spirochetes, the Lyme spirochetes, are drilling through the tissue and causing damage as they go. But then they will rapidly move throughout the entire body. They don't remain just in the skin, and they can rapidly penetrate the organs, the joints, etc., and cause a very complicated, what we call multi-system disease or disorder. One of the important things to know about Lyme disease is that it is, by definition, a chronic infection. And I want to be clear, when I say chronic, I'm talking about chronic in the absence of treatment. Treatment is generally effective if the disease is diagnosed and treated early. But in animals or humans that are not treated, the infection can persist for years and for years. And part of the reason for that is really pretty simple. It's an evolutionary based reason. So the Lyme disease spirochetes can only live in two places. They can live in a tick and they can live in a mammal. They cannot live in soil. They cannot live in the environment. So for them to maintain themselves in nature, they have to cycle between ticks and mammals constantly. And I'll talk more about that in a minute. But as a result, when a Lyme spirochete gets into a mammal, whether it's us or our dogs, it wants to stay there for as long as it can. Again, I like to think like a spirochete when I can, but it likes to stay in that mammal for as long as it can because it's hoping that another tick will come along, feed on that infected animal, pick up the Lyme spirochetes, and then transport them onto another animal and keep the cycle going. And as a result of that as well, the disease itself is actually a very slowly developing disease, but it's quite debilitating. And part of the reason that it's a slowly developing disease is that when these bacteria get into us or into our dogs or into our horses, they don't want to destroy their home. They want to live there for as long as they can, again, for the purpose of allowing time for another tick to come along, pick them up, and transport them onto another animal. We do not develop long-term immunity to Lyme disease, so you can get it over and over again. And this is very well documented. And because of that, prevention is the key in both veterinary and human medicine. So interestingly, the best data that we have that talks about the risk of Lyme disease is really generated from studies that are done in client-owned dogs or companion animals. And there's an organization called the Companion Animal Parasite Council that collects really great data that we don't really have for humans. So CAPC, as it's called, collects data from testing results that are done in dogs across the country and in Canada as well. And with that data, they generate what are called risk maps that tell us where Lyme disease is most likely to impact dogs and by extension, impact humans. So this is obviously a map of the United States. This map was generated from data collected in 2025. And states that are colored in the darker orange are considered very high risk for Lyme disease in canines. But of course, that translates to high risk in humans. States that are colored in a somewhat lighter shade of orange are considered moderate to high risk states. And then the lighter shades are states that have a lower risk. But I want to point out very importantly that even in these states that have a lower risk, quote unquote, there are pockets within those states in which Lyme disease is very abundant. So that's certainly the case in California and other places as well. So how significant is this disease? Well, Dogs that reside in endemic areas typically get tested once a year by their veterinarian for exposure to a variety of tick-borne diseases. In 2025, 535,000 positive antibody tests were reported to CAPC. CAPC only collects 30% of the test results that are run. So you can extrapolate that through, and that comes across as about 1.6 million positive dogs each year in the United States. And if you look more closely at Virginia, it's very interesting. It's clear that Virginia is a high-risk state, as indicated by these dark yellow counties. You'll notice that some counties are colored in gray. That does not indicate that they do not have Lyme disease. It simply indicates that no data was collected from those particular counties. And then other counties are shaded in a lighter shade to indicate moderate risk. So again, in 2025, there were 33,000 positive tests in dogs. Again, with 30% of data collected, that translates to well over 100,000 positives in just our state alone. These are the ticks that transmit Lyme disease. So I've placed each of the developmental stages of the tick here on a grid system so you can get perspective on the size of these particular creatures. And this is a 1 millimeter square grid system. Now, if you want to get into the details here, then the name of the tick that causes Lyme disease in our area is called Ixodes scapularis. You'll often hear it referred to as the deer tick. So the life of a tick begins when the larval stage tick emerges from the egg. The adult female will lay eggs and then the larvae emerge. And they are very small, less than a millimeter in length. What's important to know about the larvae is that when they emerge from the egg, they are infection-free. They're not carrying Lyme disease. And the reason for that is there's no ability for the female tick to transmit the bacteria through the egg to the larvae. So the only way a tick can become infected is if the tick feeds on an infected animal. And the only way an animal can become infected is if it is fed upon by an infected tick. So that's the cycle that I've been talking about, where these bacteria have to maintain themselves and cycle between ticks and mammals. Now, the larval stage tick will take what we call blood meal during this stage. So it feeds on a small mammal. It ingests blood, and then it undergoes a process known as molting. And if that larval stage tick fed on an infected animal, it will acquire the infection and then carry that infection with it through the rest of its development. So the larval stage tick takes its blood meal and it molts into the nymphal stage tick shown here, which is now the first stage of the tick that has the potential to transmit the infection to animals. The nymphal stage tick also just takes one blood meal, and then it undergoes this process known as molting. And during that molt, it develops into either the adult male or the adult female tick. You'll notice that they're anatomically very different. Adult males play no role in transmitting Lyme disease because they can only take very small blood meals. But the adult female tick has a soft palate, so to speak, or soft plate on its back. And when it feeds, it can remarkably engorge. So when an adult female tick feeds, it increases its body mass by about 500-fold. That's from the blood that it's ingesting. And I want to show you a tick on the right that's very near and dear to my heart, because this is a tick that I actually collected from my cat several years ago in Midlothian, Virginia. So Lyme disease is thriving in Midlothian, Virginia. This particular tick, my cat had come in, it was in the wintertime, so ticks feed in the wintertime. We were petting it, and we noticed a lump on the back of its neck. Of course, I get excited by that. I removed this particular tick, and to give you size perspective, I placed it on my finger so that you could see just how large the tick was. Interestingly, we took this tick into my laboratory at VCU, and we dissected out what we call its midguts, which is really its belly. And we put that into a liquid that allows us to grow Lyme spirochetes if they're present. And we were able to grow the Lyme spirochetes from this tick. So yes, Lyme disease is thriving in Virginia, and it's in my very backyard. Of course, because we were able to culture Lyme spirochetes from this tick, we paid close attention to my cat over time, and we made sure that it did not become antibody positive for Lyme disease, and it did not. which is consistent with the general idea that cats are a bit more resistant to Lyme than are humans, dogs, horses, and other mammals. I also wanted to show you a couple other just really interesting images and videos to walk you through the tick life cycle. So I mentioned that the adult female takes a very significant blood meal. This is an adult female actually in the process of mating. So this female has taken the blood meal. Interestingly, this tick fed on a black bear. My collaborators collected these ticks from black bears. So the adult female is on her back, mating with the female. This does not occur on an individual or on an animal. The ticks will feed, then they'll fall off. The mating process occurs, and then the male tick will rapidly die. The female tick will live for about a week because it has to undergo the process of egg laying, which is shown here. So this is an adult female tick held down by double stick tape. It's just starting the process of laying eggs. The eggs are being released from what we call the Jeans gland. These are the individual eggs. And what this tick is doing is arching their head over to stimulate the contractions to allow the release of the eggs. An individual tick will lay about 5,000 eggs. And if those eggs fall into an environmentally favorable spot, then those eggs can hatch and a new crop of ticks can emerge in a given area. Now, what emerges from the egg are the larval stage ticks, as I mentioned earlier. This is a brand new video we just took in my laboratory of larval stage ticks. These young ticks here are about 2 or 3 days old. And these happen to be a different type of tick. These are the ticks that are referred to as Lone Star ticks that are associated with alpha-gal syndrome and another bacteria infection called ehrlichiosis. But you can see they're quite active, quite lively, and they are very hungry. And when they get access to a mammal, they will immediately start taking a blood meal, and the process continues. I mentioned that the ticks will molt, and I wanted to show you the molt because something really important happens during the molting process, particularly when the ticks molt from the larval stage tick, that form that comes out of the egg, to the nymphal stage tick. So this is a larval tick. You'll see it has 3 legs on each side. It's opaque because it's basically shedding, so to speak. But when it emerges from the molt, from this larval molt, it develops an extra set of hind limbs. And that's really important. You'll see them here because this extra set of hind limbs allows these ticks to climb up into vegetation and get better access to larger mammals like our dogs, horses, and ourselves. If the larval stage ticks, if these ticks did not develop this extra set of hind limbs, we would probably never be talking about Lyme disease or other tick-borne diseases because they would be diseases that largely affect mice, moles, voles, and shrews. And then once a tick has emerged from the molt, it's ready to feed. They don't waste any time. They're hungry. They climb up onto vegetation and they do something called questing, where they basically sit on a blade of grass or branch. They extend out their front appendages, and when a mammal brushes up against them, they use these little hooks at the end to latch onto a mammal, and then they quickly seek out a place to feed. I think you're probably all aware of this, but just for clarification, ticks do not jump. They don't fall out of trees. They can only get access to us via direct contact. And I think that's important to remember. So I mentioned that animals play a key role in maintaining Lyme disease in nature. And certainly the most important animals are those that serve as what we call reservoirs. So a mammalian reservoir are animals that become infected with the Lyme spirochetes, and they serve as the source for infecting lots of other ticks. And some of the important reservoirs are the small mammals, a white-footed mouse, a bank vole, a pygmy shrew. We often hear about deer and the role that they play in Lyme disease, but I want to clarify something about that. Even though we refer to these ticks as deer ticks sometimes, deer are actually not really a reservoir for the Lyme spirochetes, but they are instead what I refer to as a form of mass transit. So deer, birds, other large mammals, they can become infested with ticks. And if you look at the ears of this tick, you'll see all these brown dots. Of this deer, I'm sorry. These are all ticks on the ear. They're not all Lyme disease ticks. There's a bunch of different ticks here. But as you can imagine, a deer can move over a much greater distance than a white-footed mouse or a pygmy shrew. And as a result, the deer will spread these ticks across a wider area. the adult females will fall off, the eggs will hatch, and new zones of tick density will emerge. And birds, of course, can move these ticks up and down the fly zones, and they are in part accounting for the spread of Lyme disease throughout the country. So I want to talk about the vaccine that we developed in just very general terms. Most vaccines for bacterial infections utilize proteins that are made by the bacteria itself. So the idea in general is that you take a protein made by the disease-causing agent, you purify that protein, and then you use it to immunize an individual. And when you immunize with that protein, the individual will develop antibodies to that protein. So when the disease-causing agent comes along and feeds on that immunized individual, the antibodies can attack the protein that's being made by the bacteria and prevent infection. Well, when we set out to make a Lyme disease vaccine, there were already 5 others on the market, and they were of moderate success, and we wanted to improve upon that. So we decided to make a vaccine that consists of 2 very different proteins. And the names of those proteins are outer surface protein A, OspA, or outer surface protein C. And as the name implies, these are proteins that are present on the surface of the bacteria. Well, why did we look at these 2 proteins to make a vaccine? Well, let me walk you through the tick feeding process to explain. So here comes a tick moving through the coat of a dog, and it's going to rapidly seek out a place to feed. It's going to insert its feeding structure into the tissue and draw up the blood meal. The Lyme spirochetes live in the belly of the tick, and in the belly, they are coated with this protein that I refer to as OspA. They have to have this protein on their surface to live in a tick. But when the blood meal enters in, and you can see the red blood cells here, they don't need OspA anymore, so they stop making it, and they switch to production of a protein called OspC. Now that's essential because if they don't do that, they cannot exit out of the tick, they cannot pass into the salivary glands and then pass into the mammal. So they need that protein, it's critical. So we saw an opportunity here because we had identified 2 proteins that play critical roles in Lyme disease. OspA, which is critical for the bacteria to live in a tick, and OspC, which is critical for the spirochetes to live in a mammal. So we reasoned if we make a vaccine consisting of both of these proteins, we can stimulate antibodies that can kill spirochetes directly in the tick, as well as kill the bacteria when they enter into the mammal. Now, the OspA component was very easy to produce and very easy to design because there's only one version of this protein that exists in nature. All of the Lyme disease bacteria that are out there have a very similar version of the OspA protein. What differs, though, dramatically is their OspC protein. So what I'm showing you here is a cartoon representation of what the actual structure of the protein looks like. It's called an X-ray. This is determined using X-ray crystallography. So we can actually look at all the nooks and crannies and the folds in a protein and know exactly what it looks like. So for those of us who are used to doing this, we can look at OspA or OspC and tell them apart, just like we can all look at each other and differentiate each other. Now, what I'm showing you here is several different OspC proteins, and you'll notice that they're colored differently. We colored them differently to indicate that OspC is highly diverse in nature. So there's a lot of variation in this protein. That was a problem because a vaccine needs to be designed to protect against all the different versions of the disease-causing agent that are out there. You know, we have to remake flu vaccine every year to adapt to the changes that occur. But we needed to figure out a way to develop an OspC protein that could elicit antibodies that could bind to all of the different OspCs made by all the different Lyme spirochetes that are out there. And before I tell you how we did this, I'm going to apologize to the remarkable team of postdocs, PhD students, master's students, technicians who worked on this for years, because I'm going to summarize their work in 30 seconds with all due respect. So what we did is we broke apart the OspC protein. And the reason we did this is we wanted to find the smallest piece of it that, for lack of a better term, was immunologically important. Now, what is the part of the protein that actually stimulates an antibody response, which again is what all vaccines are designed to do. And the part of a protein that stimulates an antibody response, we have a generic term for it. We call it an epitope. Well, we found the epitopes in OspC, but we found that they were very different from one OspC variant to another. So we pondered for a long time how we were going to develop a vaccine using this protein. And we finally realized that nature was not going to give us a protein that would work. So we said, all right, let's skip nature. Let's create our own protein. So what we did is we took the DNA sequences. Remember, everything's, stems from DNA. We took the DNA sequences that allow for the epitopes of each of these different OspC proteins to be made, and we created a new piece of DNA, a new gene. Now, this new gene is not OspC, but instead it's a new protein designed in the lab that has the best parts of many different OspCs joined together. And why do we do that? We wanted to make a protein that could stimulate production of antibodies that could protect against Lyme spirochetes that are making any of these different OspC proteins. And that worked. It was very effective, and it led to Vanguard's CR Lyme. CR Lyme, as was mentioned, came on the market in 2016. It's approved by the USDA. It's gone through extensive safety trials, and it's now the most widely used vaccine in the United States and Canada. So without getting into a lot of details about our vaccine, I want to talk about whether vaccination makes a difference, because I often get asked this question. Well, it's great that you have a vaccine, but does it really matter? What does it really do? Well, we have good information to look at this in our canine friends. So what we're looking at on the left is the state of Massachusetts. And this is one of those companion animal parasite council maps I mentioned earlier. You notice the whole state is colored in dark orange. All of Massachusetts is high risk for Lyme disease. And that hasn't changed between 2016 and 2025. But why am I showing you Massachusetts? Well, Massachusetts is a state that adopted vaccination for Lyme disease in dogs early on. They're ahead of many other states. And, did vaccination help? Well, in 2016, 1 in 6 dogs were testing positive for Lyme disease in Massachusetts. But in 2025, that number has decreased to 1 in 10. That's a very significant decrease in the number of animals testing positive. Over this same time period, the number of humans that have tested positive has skyrocketed. And the number of animals that test positive for other diseases transmitted by these same ticks have also dramatically increased. So this is one example where we can look at the data and say, yes, vaccination makes a difference. And if we can improve the vaccination rate, we can improve the health of our companion animals, and ultimately we can do that with a vaccine when it moves into humans. Now, I'm just going to close and tell you a little bit about what we're doing to develop a human vaccine. We developed a modified form of CR Lyme. And when I say modified, we had to design a new version of the vaccine to be specific for use in humans in a variety of different ways, which I won't elaborate on. But we did that, and we tested that new vaccine in mice. And using the vaccine formulation, we found that we could protect 90% of mice from infection with the Lyme spirochetes. That's a great number, especially after only 2 doses of vaccine. But in order to move a vaccine into human use, you have to demonstrate its efficacy in a non-human primate model. Now, we are all very sensitive to working with non-human primates, and we certainly consider that very carefully, but it's a required step in order to get FDA approval to advance a vaccine into humans. And I will just tell you, we did a very extensive study with Tulane Primate Center. I'm not going to go into any details, but what I will note is that we gave 2 doses of the vaccine 1 month apart. So this is very typical for vaccine. It's called a prime and then a boost schedule. You hit the first dose and then the second dose. And then we fed infected ticks on the rhesus macaques several months later. And we asked via a variety of mechanisms, did the vaccine provide protection? And the answer was that we achieved 100% protective efficacy with the vaccine. So this was fantastic. NIH became very interested in this, and they solicited us to work with them to develop what's called a customized product development plan. And what they do is they hire a team of external experts to get together, analyze your data, decide whether it warrants moving forward, and if it does, what exactly do you need to do to advance it to phase 1 clinical trials? So we're really grateful that that occurred and that was completed last year, which was very important because timing of that was really everything. But with that, I'm going to summarize and tell you what I've told you today. So Lyme disease is an increasingly important disease, and it continues to spread. It used to be largely a disease of the Northeast and the upper Midwest. That's no longer the case. I was just out in Iowa and talking with vets who are seeing large numbers of antibody positive tests out there. It's spreading dramatically. Vaccines that have shown you have proven effective in dogs, and they play a key role, and they can play the same role in humans. But it's important to note that Lyme disease is not the only tick-borne disease. There are others that you've probably never heard of that are equally important and even more problematic in terms of human medicine. So a Lyme vaccine will maybe potentially solve the Lyme problem, but not the tick-borne disease problem. I've told you about a unique technology we developed at VCU called chimeritope technology. That's chimeric epitope proteins, those unique OFC proteins that we made. CR Lyme is the first vaccine to utilize this technology. And when we started, there was very little work being done on this chimeritope approach. But if you look now at the vaccine journals, it is almost exclusively utilizing this approach, which was pioneered at VCU. So we're using chimeritope technology to develop additional vaccines for additional diseases, as well as using it to develop diagnostic assays. And really, this technology can be applied to any infectious disease. And we are working aggressively to advance a human vaccine, although the current climate has certainly placed some limitations on that. And with that, I want to close and just thank the current composition of my lab. Again, these are really brilliant people. These are, people from the state of Virginia and elsewhere who are learning about science. They're dedicated, they work hard, and their ability to do work is strictly dependent upon obtaining resources through funding, etc. So with that, I will stop my share, and I'd be happy if there are any questions to address them for you. Greg, I think you're muted.
Unmute. There we go. Thank you. Normally that doesn't happen. But that was fascinating, very interesting, and Evelina, the same information that you provided as well. Richard, I have some questions for you as we wait to see if anybody wants to put any questions in the chat function.
Sure.
Please do so. But here's my question for you. You talk about the clinical trials last year. I'm guessing that was for human clinical trials. Where does that stand? How are the results, and how soon are we talking? 2 years, 5 years, 10 years for a human vaccine?
Yeah, so first of all, the clinical trials that you're referring to are actually done on a different vaccine. It's not the vaccine that we are working on, but I'm glad to see any vaccine advance because we certainly want, different options in the marketplace. But let me tell you about that particular vaccine. That was a vaccine that was originally developed by a company called Valneva. Valneva then partnered with Pfizer, and they worked together to advance their vaccine. Now, you may remember that I mentioned this protein earlier in the talk called OspA. Well, OspA is what that new vaccine is comprised of. So Pfizer/Valneva just completed phase 3 clinical trials. And in those phase 3 clinical trials, they delivered 4 doses of the vaccine in the first year of use and achieved 73% protective efficacy. So, we're really interested in continuing to advance other options because one of the concerns I have about that particular vaccine is compliance with a 4-dose or a 4-stick regimen over a year. We know that there's been vaccine hesitancy and compliance with that, that type of a protocol. I have some concerns about it. But having said that, I think the vaccine is still a valuable, addition to our arsenal for preventing Lyme disease.
So let me ask you, Richard, and also Evelina, how difficult is it to obtain funding these days?
Yeah, I guess I'll tackle that one because certainly a lot of firsthand experience with that issue. Well, so it kind of depends upon what you're working on. Vaccines have taken a hit. It's a really unfortunate scenario right now, particularly certain types of vaccines, which have fallen out of favor for reasons that I won't bother to try and articulate today, with the National Institutes of Health and the Department, and et cetera. So. Funding is a challenge in the vaccine world if you're trying to acquire it through the National Institutes of Health and other federal agencies. But as Evelina had pointed out earlier, we have obtained a lot of our funding either through the VCU Tech Transfer Program, through the Virginia Innovative Partnership Corporation, through the Commonwealth Health Research Board, through commercialization funds that are available through VCU, and also, of course, through industry. So we have worked with lots of companies in various different capacities to bring in funding to support the development, not only of our Lyme vaccine, but of other products that we're currently advancing. So, I have to certainly acknowledge the importance of private donations and foundation support, which have played a key role in our ability to advance these things without NIH funding.
So here we have a question from someone on the on here today. Nora wants to know, what are your thoughts on Lyme possibility being a STD or transferred from mother to child? Do you think Lyme could have any relationship to the proliferation of autoimmune diseases?
Yeah, that's a great question. We actually did a study on this in canines several years ago. Now, of course, dogs are not humans, so let's keep that in mind initially. But what we found is we were analyzing a pregnant Rottweiler that had Lyme disease, and we were able to enter into a collaborative arrangement with the veterinary clinic and the owner of that dog to assess not only the mom through the process, but also the pups. And we found that there was actually no evidence of transplacental transmission. Now, of course, animals differ, right? But there was some concern that this could be going on in human medicine, that there could be transmission, but the evidence for it is really very, very weak. So, my personal conclusion and opinion is that we do not need to be concerned about transmission in utero, but we do need to be very concerned about the health of the mom who's experiencing Lyme disease during pregnancy. So that's kind of the state of where we're at in our understanding of that specific question.
So Richard, and maybe this is for you also, Evelina, but what opportunities does the Richmond region or Hampton Roads regions offer to researchers? How can we help?
Evelina, would you like to take that
The
one?
the your Greg, you mean the innovation partners? Yes.
Yes.
Well, yeah, I mean, we actually collaborate actively with Lighthouse Networks here in town, with Activation Capital, with the Angel Networks in the Commonwealth. And the collaboration is mostly for spinning off and supporting new ventures. Because it takes a village to grow a new venture. It takes a lot of effort from different organizations, not only from the university. University now would do what we can, but we need to really have the network to help the company with the next steps. Space, funding, providing the right management team. So as I said, it needs really collaboration of everybody in the region to get successful startups off the ground. Another opportunity to collaborate is in enhancing university industry collaborations and engagement, creating public-private partnerships, and especially nowadays when the federal funding is either decreasing or not enough to support different inventions such as vaccine development, as Dr. Marconi mentioned. So yeah, there are a lot of opportunities.
And Evelina, you raised an interesting question I was going to ask in terms of, can either you and or Dr. Marconi talk about how the impact of federal funding has been on university research?
I mean, I'd be happy to start with that real
Yeah.
quick. So we have in my lab, we basically kind of saw this coming. So we worked very hard to aggressively prepare for the change in the funding landscape. And I will tell you that one of the wonderful things that Virginia Commonwealth University has done for us is that back in 2016, when the vaccine launched, they agreed to return their share of the royalties into a research fund for us. And the importance of that is it allowed us to work on vaccines and other products for which we did not have federal funding. Now, in 2016, I don't think we would have envisioned how important that was, but now it's quite important. So we've prepped for it and we've tried to tap into a diverse array of funding opportunities outside of NIH. But we still pursue NIH, of course.
Great. Well, thank you. The time has come. It's 1 o'clock. Let me, we really appreciate it. Let me just real quickly, let everyone know what's coming up next. Just a reminder, go to our website to find out what our major initiatives are, information about what's happening in the General Assembly related to digital infrastructure ecosystem. Also, as a reminder, the recording for this and any of our webinars will be on our website. This recording will be on our website sometime later this afternoon. Please share it with others. I'll send out a reminder email to let everyone know. But also, all of our webinars for the past since 2022 are all on our website. So we have had a lot of very, very interesting topics over the years. So give it an opportunity to go look at those. And reminder, just on Wednesday, May 6th is our next Innovation Spotlight webinar. Hope you can join us for that. I'll be sending out information about that here in the next week or so about the topic. And again, thank you, Richard and Evelina, for being speakers today. Very fascinating, interesting conversation about Lyme disease and the bigger issue of what universities are doing to be really powerful in terms of partnerships in our region. And also, thank you to Horgan for its sponsorship today and for its corporate support as well, Brian. We appreciate it. And thank you, everyone, for being on here today. We look forward to seeing you next month on Wednesday, May the 6th. Have a great afternoon. Thank you so much.
Thank you.
Thank you.